Next time we have the three variables of mitochondria.
Good day, ladies and gentlemen. It is my honor to welcome you to today's discussion on a fascinating aspect of genetic transmission, the extrachromosomal inheritance. In the realm of genetics, we're accustomed to dance of trait passed down through the chromosomes, but today we delve into the realm beyond, where genetic information takes a new dimension. I'm Cassandra Eliza Tucaro. Welcome. Delayed extrachromosomal inheritance refers to the transmission of genetic information through mechanism other than the standard DNA sequence within the chromosomes. This phenomenon involves the inheritance of genetic traits that are not encoded in the chromosomal DNA but instead are carried by the extrachromosomal elements such as plasmid and mitochondria. These extra chromosomal elements can replicate independently of the chromosomal DNA and can be passed on to offspring through mechanisms like cell division or maternal inheritance. One well-known example of delayed extrachromosomal inheritance is the transmission of mitochondrial DNA or mtDNA from the mother to her offsprings. Mitochondria, the energy-producing organelle in cells, contain their own small circular DNA separate from the nuclear DNA. This mtDNA is inherited exclusively from the mother, and the mutation in the mitochondrial genome can lead into various inherited mitochondrial disorders. Understanding delayed extrachromosomal inheritance is important in the study of genetics and inheritance patterns, as it adds complexity to the mechanism by which genetic information is passed from one generation to the next.
Good day everyone, I'm Jacqueline Tiamon, a second-year college student taking Bachelor of Science in Agriculture at Laguna State Polytechnic University, Sinaloan Campus. Extrachromosomal inheritance involves genetic materials outside the cell chromosomes, primarily found in organelles like mitochondria and chloroplasts. Three key criteria define this mode of inheritance. First is autonomy. Extrachromosomal elements such as mitochondrial DNA have their own replication and maintenance system independent of the nuclear genome. This autonomy allows them to evolve separately, influencing mutation rates and evolutionary paths. Maternal inheritance. Extrachromosomal elements are predominantly inherited from the material's lineage due to the contribution of cytoplasm containing organelles from the egg during fertilization. This maternal transmission patterns has implication for disease linked to mitochondrial DNA mutation. Their unique genetic system. Organelles like mitochondria and chloroplasts possess unique genetic codes and systems for the gene expression distinct from the nuclear genome. This difference reflects their evolutionary history and function specialization.
Hi, I'm Dana Flora Concern and I'm here to talk about fascinating topics in genetics, which is the extrachromosomal inheritance. But first, let's break it down into simple terms. Now, in simple terms, imagine your body is like a big book, and each page of that book contains instructions for different traits, like your eye color and your height. These instructions are written in a language called DNA, and they are organized into chapters called chromosomes. Usually, when you inherit traits from your parents, you're getting pages from their book or chromosome, usually shuffled around like a deck. This is what we call normal inheritance, but sometimes things get little more interesting. How extrachromosomal inheritance happens when there's something extra going on outside of that chromosome. It's like having bonus chapter in a book that aren't part of the main story. The third criteria for the extrachromosomal inheritance, the non-segregation. Imagine you have a deck of cards and presenting the eye color and the hair texture. In normal inheritance, when you shuffle these cards and pass them on to your children, they get a mix of traits from both parents. But with non-segregation, it is some of these cards stick together and they don't get shuffled properly. So instead of getting mixed trait of the parents, they end up getting a bunch of cards that are all the same. So how do we know if something is inherited extra chromosomal? Well, there are few things to look for. One example of the non-segregation is certain type of plants where traits are from variegated leaf patterns don't separate out as expected. Instead of getting some leaves with patterns and some without, you might see a whole bunch of branches with consistent patterns. Fourth criteria is the non-Mendelian segregation. Now, remember Gregorio Mendel and his famous pea plants? He discovered that some traits don't follow the predicted pattern of inheritance, like the dominant and recessive trait. But with non-Mendelian segregation, traits don't follow these neat rules. Instead, you might see variation that don't fit into Mendel's categories. Example, in certain types of chicken, feather colors don't follow the typical dominant and recessive pattern. Instead, you might see multiple feathers often expressed together, creating a unique and unexpected look. Last criteria, which is the infection-like transmission. Now, this one is a bit tricky to imagine, but think of it as like passing down a trait without the usual genetic pathway. It is almost like the trait is contagious, spreading from one generation to the next without involving the chromosomes directly. Example of infection-like transmission is seen in a certain types of fungi that can pass traits like a drug resistant from one generation to the next through a process called cytoplasmic inheritance. Instead of relying on the nuclear DNA, their traits are transmitted to other cellular components. That is all. Thank you for watching.
Hi everyone, my name is Juladriel P. Basco from BSA 2C. For today's video, we are going to discuss about plasmid of extracellular origin. Plasmid can only multiply inside the host cell. Plasmid naturally exists in bacteria cells. They also occur in some eukaryotes. Often, the genes carried in plasmid provide bacteria with genetic advantages such as antibiotic resistance. Here we have an example of plasmid, an episome. Episome is a special type of plasmid that remains as part of the eukaryotic genomes without interrogation. But if a bacteriophage and chromosomes collide, it can replicate independently to the host cell. Next, we have infectious heredity. When a bacterium containing a plasmid reproduces, it can pass the plasmid to an offspring or daughter cells during cell division. If the plasmid carries beneficial traits, these traits can spread within bacterial population. The transfer of plasmid is divided to three several mechanisms. Number 1. Conjugation. Conjugation is a cell-to-cell transfer of plasmid via proteinaceous bridge between donor and recipient cell. Number 2. Transformation. Transformation uptakes of free plasmid DNA from environment by recipient cell. And lastly, transduction. Transduction is a plasmid of bacteriophage viruses that affects bacteria during infection.
Good day everyone, I'm Rabansa Rosanti from BSA2C handled by Miss Maria Lerlin Logit, and in this video, I will tackle about extra chromosomal inheritance and cytoplasmic inheritance. So that is the transmission of genes that occurs outside the nucleus. It is found in most eukaryotes and commonly known to occur cytoplasmic organelles, such as mitochondria and chloroplasts, or from cellular parasites like viruses and bacteria. It is occur when phenotype results from genetic influence other than the transmission of parental genes or maternal inheritance located on chromosomes in the nucleus. The cytoplasm is the cell substance between the cell membrane and the nucleus containing cytosol, organelle, cytoskeleton, and various particles. And in chloroplast, it is the process of extrachromosomal inheritance. The structure, the pigment, and the enzymes can be all affected by alteration and changes. And we have the three variables of mitochondria. It is the nuclear, neutral, and expressive. The nuclear is the variable that is chromosomally determined trait, and the neutral is the variable that indicates an extra chromosomal inheritance or disappearing of traits through times. And suppressive, this variable acts as dominant trait.